Okay. All right. Well, hello, everyone who's here physically and also who's present on Zoom. My name is Sarah Smith, and it's my pleasure to introduce our seminar speaker today, Dr. Randie Bundy from the School of Oceanography at the University of Washington. Dr. Bundy leads a research group at the UW focused on the biogeochemical cycling of trace metals, iron, copper, nickel, and cobalt, for example, in the marine environment. And Dr. Bundy obtained her undergrad degree in environmental systems with a specialty in earth science and chemistry from UC San Diego. She stayed in San Diego and obtained her master's and PhD from Scripps Institution of Oceanography in the lab of Professor Kathy Barbeau. She followed this with a postdoc at Woods Hole, so kind of hitting all the big institutions, East Coast and West Coast, where she was co-advised by Dan Rapetta and Max Sato. And shortly after starting her lab at the University of Washington, Dr. Bundy was awarded as a Simons Early Career Investigator in Marine Microbial Ecology and Evolution, which is a very significant and very prestigious award, kind of kickstarting her research program at UW. She's the lead PI or co-PI on other NSF-funded projects focusing on different aspects of trace metals, like complexation by siderophores. Randie has extensive field experience and has participated in, from my count on her CV, 24 research cruises of 2 or more days, adding up to a total of 485 days at sea, or over an entire year and 4 months. So that's a pretty significant amount of time spent on a ship. Her longest cruise is one of, I believe the most recent, when she spent 55 days at sea investigating hydrothermal plumes on a cruise called Plume Raiders. From which I believe she'll share some of her data with us during her presentation today. Welcome, Randie, virtually to Moss Landing Marine Labs. We're so excited to hear about your work today.
Thank you. Thank you so much, Sarah. I'm really excited to be here and to chat with you all about the work that's been going on in my lab. And as Sarah mentioned, specifically, our lab focuses on studying trace metals in the marine environment. And today I'm going to talk to you about the microbial production of organic compounds or organic ligands and how they impact the cycling of iron in seawater. And this is, of course, all being done by hardworking members of the Bundy Lab, some of which are listed here just for the projects I'm going to talk about today, as well as some other collaborators at Scripps, including my old PhD advisor, Kathy Barbeau, and Lauren Mank, who used to be in Kathy's lab as well and is now a postdoc, and then 2 undergraduates that have been working in my lab, Patrick Monreal and Dylan Vecchione. So why are we so interested in studying iron in seawater? Well, it's a really important micronutrient for both phytoplankton, bacteria, and archaea in seawater. And it's the 4th most abundant element in the Earth's crust, but it's very, very low in seawater. And part of that is due to its poor solubility, which I'll talk a little bit about later. And I like this figure because it summarizes different elements and their kind of average concentration in seawater relative to what's required for growth by kind of the average large phytoplankton cell. And we can see that iron falls a little bit to the left of this kind of dashed line here, showing that it's relatively depleted in seawater relative to what's required for phytoplankton growth. And because it falls in this kind of light gray shaded area, that means that it's actually about 100 times more depleted in seawater relative to what's required for growth. And because of that, it's often a limiting nutrient in many areas in the ocean. All of these little dots on this diagram where the background colors are nitrate concentrations in surface waters. Each dot represents a different bottle incubation experiment where the researchers investigated which nutrients were limiting overall community primary production in that particular region. And so we can see that there's a lot of red dots, and that signifies areas where iron was the limiting nutrient for growth. And this happens in a lot of these regions where nitrate and other macronutrients are sufficient for phytoplankton growth, but iron is super low, partially because, again, of its poor solubility, but also because many of these areas are pretty far from margin sources of iron, which is where iron is really abundant in Earth's crust. And these are all areas that we call high nutrient, low chlorophyll regions. So these are areas where there's kind of lower than expected chlorophyll, because there's high macronutrients. So there's definitely a lot of primary productivity going on, but iron is kind of limiting that biomass to some level since it's so low. And as I mentioned, iron is really low in these regions, not only because they're kind of far from margin sources of iron like dust or sediments, but also because iron itself, in terms of its inorganic solubility, it's very poor in oxygenated seawater. So if it was only inorganic iron in the ocean, where we have dissolved iron up here on the x-axis and nanomolar concentrations, its profile on average would kind of look like this green dashed line. So it'd be kind of tens of picomolar concentration if it was just inorganic iron. Fortunately, the iron profiles in the ocean on average kind of look like this red one here, and that's a lot higher than we would expect just based on its solubility if it was all inorganic iron. And so this is good news for organisms in the ocean that need iron, because at least there's a little bit more of it than we would expect. And the reason for this is because there's a bunch of organic matter in the ocean, and much of this organic matter has the ability to bind or complex to iron, and this actually enhances its solubility. So in fact, about 99% or more of the dissolved iron in seawater is associated with some kind of organic molecule, and we call these ligands. So these are organic compounds that bind to metals. And here I'm showing kind of average profiles of two operationally defined classes of ligands, which I'm going to talk a lot more about later, called L1 or really strong ligands and L2 really weak ligands. But you can see that their profiles kind of look like dissolved iron, and they're also present in kind of nanomolar type concentrations. So these ligands really enhance the overall solubility of iron. And because of this really important control that these organic molecules have on the solubility of iron, we actually have realized in the past several decades that we have to really understand the sources and sinks of these ligands themselves in order to understand distributions of dissolved iron. And this has been really nicely shown with several biogeochemical modeling studies, where we're comparing different models that both contain an active parameterization for organic molecules that bind iron, or these ligands, and other models that don't contain these ligands. And so there's been the advent of tons of measurements of dissolved iron in seawater with the International GeoTraces program. And this is one example of such a transect in the Atlantic Ocean that goes down kind of a slice of the middle of the Atlantic. And here we have the distributions of dissolved iron measured on that transect in the upper 500 meters on this panel, and then the lower 1,000 to 5,000 meters on this panel. So these are our observations of dissolved iron. And if we first look at one model that models the iron cycle but doesn't model organic ligands, we can see that the model output doesn't match our observations very well, particularly in the upper 500 meters, where we see a lot of green colors here. And in the observations, we see a lot more of these kind of like blue, purple colors. But if we look at one of our models that explicitly includes a ligand cycle embedded within the model, this output looks a lot more similar to the observations that were observed along this transect. And in fact, we now know that we can really only model iron distributions in the ocean if we actually include an explicit kind of dynamic ligand cycle in the model as well. And not only is including ligands in the model important, but the fact that they're there can have long-lasting impacts, and even more so than just kind of getting the distribution of dissolved iron in the ocean correct. In this particular example, it was pointed out that if you change the concentrations of dissolved iron binding organic ligands in the model, it has significant impacts on atmospheric CO2 in the model, where if you have or double the ligand concentrations in the model, it modifies CO2 greater than if you were to, say, double the dust or hydrothermal inputs of iron itself in the model. And that's because these organic ligands control the solubility of iron so much that even if we're kind of increasing sources of iron to the ocean, we can only have as much iron in the ocean as we have ligands. And so they're really important for kind of controlling this overall inventory of iron in the ocean, and for connecting the iron cycle to the carbon cycle and even atmospheric CO2. And so we now include ligands in our kind of state-of-the-art biogeochemical models for iron cycling. And this is a schematic of one such model here. And there's kind of two major places where ligands play a big role. And I'm going to kind of highlight two vignettes in our lab where we're looking into these two aspects of ligand cycling in more detail. So the first is if we think about processes going on in the deep ocean, these models have this kind of scavenging rate of iron set by this variable ligand concentration. So depending on how many ligands are present in the deep ocean and their sources and sinks, we can kind of set these deep ocean iron concentrations in the model. The other way is we have ligands that are produced by microbes in surface waters. And these ligands have also a strong impact on the kind of inventory of iron, longevity of iron in the surface, and also its recycling and bioavailability. However, these ligands are hard to understand because they are just a very small part of this really super diverse organic matter matrix that exists in seawater. If you remember from the average profiles of ligands that I showed you, they're in kind of nanomolar concentration ranges, whereas dissolved organic carbon in seawater is micromolar ranges, and it's super complex, it's very diverse, lots of different functional groups, lots of different physical sizes. So we're kind of looking for this small component of an already really heterogeneous pool. So it's a little bit a needle in a haystack problem, and we have to bring a lot of geochemistry as well as trace metal chemistry analytical tools to bear on this problem. So I'm going to talk to you a little bit about some of the methods that we use then for kind of disentangling what these actual ligands are from this complex organic matrix. So one method that we've been using for a long time and was kind of the initial measurements that were used by the GeoTraces program are these voltammetry methods. And these methods involve essentially doing a titration. So you take your seawater sample, you put it into several different aliquots in some little vials, and then you titrate the natural organic compounds that are present in your sample with additional added iron. So you're kind of titrating any natural organic compounds with iron so that you can have all of your iron bound by organic matter in your solution. Then we add a known competing ligand. So this is a ligand that we know binds iron, we know how strongly it binds to iron, we know a lot of things about it, we know its kinetics, etc. And we kind of set up this competition between the natural ligands and this known ligand, which we call salicylaldoxamine, or SA for short. Then we let that equilibrate, and then we measure our iron that's associated with our competing ligand after some amount of time when it's fully equilibrated. And because we know how strong our competing ligand is and how tightly it binds to iron, we can then calculate the concentration of the natural ligands as well as the binding characteristics. So this gives us kind of a total concentration of iron binding ligands in our sample, their binding strengths, and it allows us to kind of operationally define classes of ligands based on that binding strength. And we usually classify them simply as either L1, which are super strong ligands, or L2, which are weaker binding ligands. This is a nice method because there's very little manipulation to the sample. It can be done on relatively small volumes, like 100 to 150 mils per titration. So this has been used widely by the GeoTraces program. However, it doesn't give you any information about the actual identity or masses of these ligands. And so we also use some other methods that we've been developing over the last decade or so. And this is methods that involve a solid phase extraction step followed by various mass spectrometry techniques. So for this, we take our filtered seawater, we pump it onto a column and do a solid phase extraction. All the organics that are bound to iron are retained on the column, and then we elute that into a smaller volume. And then to measure the ligands, we first inject this sample onto a column. Using liquid chromatography, we separate the compounds, and then we connect this column to two different mass spectrometers. The first is an ICP-MS, and this actually allows us to determine the retention times of compounds that contain iron or other metals of interest, as well as to quantify them. And then we do kind of a second step where we use the same chromatography, but we connect it to an electrospray ionization mass spectrometer. And this allows us to kind of softly ionized compounds. It leaves the compounds intact, and we can determine the mass to charge ratios of those compounds. So we kind of have these two steps here where we can both quantify and identify metal containing compounds, as well as determine what their mass is and hopefully some other characteristics about them. So these methods allow us to measure the concentrations as well as kind of the identities of these iron binding ligands. We can derive chemical formula for these compounds and hopefully identify the ligands. And this method generally targets microbially produced ligands. That's how we've kind of developed them. The downside to this is that we need a lot more volume. When I started doing this work as a postdoc, I needed like 800 liters, and now we've gotten it down to 4 liters. So we're getting a lot better, but it's significantly more volume than these kind of voltammetry methods. So these are the two methods I'm going to be talking about today for how we're trying to kind of understand these iron binding ligands in seawater. So using these voltammetry methods, which again have been used widely by this International Geotraces program, which has set out to map trace metals and their isotopes and their chemical speciation throughout the global ocean. These are many of the stations where ligands have been sampled in this program. And these measurements have been made using voltammetry, or the method where we're just kind of understanding the binding strengths and classes of these ligands. And from these methods, we've seen, this is just one example from this transect here in the South Pacific. We see that both strong and weaker ligands are present throughout the water column, throughout the global ocean. And they're in concentrations that are kind of nanomolar, and they look very similar to dissolved iron concentrations. I'm going to focus today on these strong ligands, which I'll explain a little bit later. So these are the ones that we call L1 type ligands. And the reason I'm going to focus on these is that they have the strongest relationship with dissolved iron. So if we take a lot of the data that's been collected from geotraces, and we have it on this plot here of dissolved iron versus the strong ligand concentration, we see that there's this really nice, strong correlation between dissolved iron and strong ligand concentrations. And the same is not true if we include total ligands. So if we include kind of the strong and the weak ligands, we get a much weaker relationship between dissolved iron and the total ligands. So we know that the strong ligands are really kind of instrumental for controlling the dissolved iron distributions. And currently we actually only model these L1 ligands. So these are the only types of ligands that we include in our biogeochemical models for dissolved iron. So I'm going to focus on them. And these ligands are really interesting because we're not entirely sure what all of them are, but we think that one really promising candidate is siderophores. And siderophores are really interesting because they are compounds that are low molecular weight. They're produced by bacteria, and they have a super, super high affinity for iron. And they're produced in the soil environment by bacteria that want to kind of solubilize iron from the soil. And in the marine environment, they can be produced intracellularly, and then they are extruded into the marine environment. They bind to iron 3, and then they can be taken up by specialized transporters across the outer membrane, brought back inside the cell. The iron is released and the siderophore is released, and then the siderophore can be used again and kind of recycled in this manner. There's a couple different kind of broad classes of siderophores, but some of them sort of look like this, and they can actually diffuse away from the cell. But some of them look like this one here, where they have this long little chain. And in this case, this chain kind of partitions into the outer membrane, and microbes can use them as kind of like a little fishing pole for iron, where they just kind of like try to grab it from the marine environment, but it's still connected to the cell. So these were first identified in the terrestrial environment, but we think that they could definitely be present in seawater. And the reason for that is that the binding strengths of these compounds are really similar to the binding strengths that we've measured for L1 ligands using our voltammetry methods. And then in incubation experiments in the field, where we see these iron limiting conditions for the microbial community, we see that L1 ligands are produced. And this makes sense because the biosynthesis genes for these siderophores are regulated by the ambient iron concentration. So if there's low iron, the bacteria tend to kind of turn on the biosynthesis genes for siderophores. So the fact that we're seeing ligands being produced in low iron conditions that have similar binding strength to siderophores seems really promising. And as I mentioned, we're getting a lot better now at our mass spectrometry techniques, which is how we can identify siderophores in seawater. And so we've actually been able to observe them now in surface waters in the marine environment. And what we know so far, there's been very few studies that have measured these directly in seawater because it's such a difficult task. But this is just one example where we know that siderophores at least comprise some portion of the L1 ligand pool in seawater. So this is a profile at Station Aloha. This is dissolved iron in these open circles. And then this is kind of the total strong ligand concentration in black that I measured by voltammetry techniques. And then if we look at the amount of iron binding ligands that we were able to extract with our solid phase extraction technique, the profile looks very similar to the total ligands, but the scale here is a lot lower. So we were only able to capture about 10% of the ligand pool on just our solid phase extraction column. And then in terms of all the siderophores that I could actually definitively identify in the sample, we get kind of picomolar concentration. So in general, we're able to capture some amount of this kind of total strong ligand pool in seawater with these like new mass spectrometry methods, but we can only identify a subset. But we know at least that siderophores are there. They're definitely present, but we're probably either missing some other siderophores that we're not capturing with this technique, or there's other unknown ligands that have these strong binding strengths. So despite the fact that we haven't been able to identify all the compounds that are part of this kind of strong ligand pool, we do know that they have a significant impact on dissolved iron distributions in the ocean. And two of the things that I'm going to kind of highlight today are related to, first, kind of the resonance time of iron. So in this example, this was dissolved iron distributions observed in the southern Pacific along the southern East Pacific Rise. And we saw this surprising plume of dissolved iron emanating from the ridge crest. And I'm going to talk a little bit more later on about how ligands might be playing a role in kind of stabilizing and transporting this dissolved iron thousands of kilometers away from this hydrothermal vent. And then the other aspect of iron cycling that I'm going to highlight where siderophores and strong ligands in general play an important role is with the bioavailability of iron. So we know that organically bound iron has a very different accessibility to the microbial community than simply inorganic iron, where uptake of inorganic iron is generally much higher across all species compared to different forms of organically bound iron, including siderophore bound iron. For these two are examples. And that's because you need specialized cellular receptors in order to take up these organic forms of iron. So these strong ligands kind of have this outsized influence on the bioavailability of iron. So the research questions that I'm going to discuss with you today are how and why are microbes producing these organic ligands in the marine environment? And in general, how are these ligands kind of impacting the bioavailability and reactivity of iron? So the first story that I want to talk to you about is work that's being done by one of my graduate students, Ji-Woon Park, who's pictured here. And we've been looking in the marine environment about how siderophores can impact iron bioavailability by solubilizing iron from different minerals. And as I mentioned, this is kind of how siderophores work in the terrestrial environment. So we've been interested to explore this in the marine environment, where siderophores kind of promote dissolution of iron from soils. And in fact, if you use siderophores that we know are made by marine bacteria, such as ferrioxamine B, we can see that iron dissolution from dust is really enhanced when a siderophore is present, as opposed to just kind of simple reductive dissolution of iron from this mineral. So in theory, we think that siderophores should be kind of used in a similar fashion in the marine environment as they might be in the terrestrial world. And so one really cool model system that has been developed by Lauren Mank and Kathy Barbeau, when Lauren was her graduate student at Scripps, was using Alteromonas macleodi, which is a copiotrophic marine bacteria that kind of disproportionately impacts the processing of organic matter and nutrients in the marine environment. It has a lot of different genes for processing all types of organic matter. It's usually present when there's kind of a pulse of nutrients or carbon. And what Lauren noticed is that this particular organism has siderophore biosynthesis genes, and she thought that it might be producing this siderophore called petrobactin. So she sent us some samples of her wild type culture, and we were able to identify on our mass spec that it was indeed producing this siderophore, petrobactin. And so what Lauren wanted to know was how this organism was using that siderophore. And so she decided to make a mutant where she knocked out the biosynthesis genes for making this particular siderophore. She then sent us some of the culture medium from this mutant, and it was clear that it was no longer making petrobactin. So that was great. So what she wanted to do was then follow up with some experiments with both this kind of wild type and this mutant and see if it could access different types of iron from the marine environment when it no longer had the ability to produce this siderophore. So what she did was she had two separate cultures that she was working with. She had her mutant that had none of the siderophore biosynthesis genes, as well as her wild type here in the blue. And then she gave it a bunch of different kind of refractory iron sources and also just inorganic, very super bioavailable iron as kind of a control. So both the mutant and wild type could grow just as well on the inorganic iron. But then when it was given kind of more refractory iron sources, including kind of colloidal iron and different types of particulate iron, the strain that didn't have the biosynthesis genes, or her mutant, was not able to grow as effectively on those sources. And in fact, its growth rate was diminished as well on these different particulate iron sources. So it was clear that A. macleodi was using the siderophore petrobactin in order to kind of access these different particulate forms of iron. And what was really interesting is that when we looked in our data sets that we've been collecting in the North Pacific along a couple transects here, which I'll go into a little bit more detail in a while, we actually have identified this siderophore petrobactin. Petrobactin is kind of a unique siderophore because it's photochemically reactive. And so we mostly saw petrobactin present in deeper waters, kind of below 100 meters or so, but we also found its photoproduct or its derivative in surface waters as well. So this suggests that this is kind of a nice relevant model system on how heterotrophic bacteria could be using siderophores in the marine environment. And in fact, this particular compound is definitely relevant in the marine environment. And not only was this particular siderophore found to be present in the North Pacific, but we saw a lot of other compounds, and many of these were kind of present in this region where we know there's a lot of dust or mineral inputs of iron. So as I mentioned, we've been doing some work in the North Pacific, and we've gone on several cruises now with the Simons Foundation, going from the subtropical gyre to the subpolar gyre along this kind of gradient in iron and nutrients. We cross this transition zone that's kind of in between the two gyres. And this is a region where we have often a lot of dust input in the spring. And across this transect, we saw relatively high concentrations of siderophores compared to what we've seen just at Station Aloha. Again, in kind of these picomolar ranges in terms of what we can identify. And when we looked a little bit more closely at this data, we have two cruises that I'm including in this study here that my student Jiwoon just recently submitted to LNO. But if we look just in surface waters, the siderophore concentrations were relatively low on our first cruise, which was kind of really early spring before this pulse of dust is introduced into the transition zone region. But then the siderophore concentrations were quite high on our second cruise, which coincided with much higher dissolved iron concentrations in the transition zone shown here in blue. And also it was kind of the peak of dust input to that season. So it's kind of this like May, June timeframe. And this is showing satellite data where the darker colors are showing more dust input to the region. So we saw not only the presence of petrobactin, which we know heterotrophic bacteria could be using to access mineral forms of iron, but we also saw high siderophore concentrations in this region where we know there was high dust input. And we've done some incubation experiments as well, where we've given the in situ microbial community different sources of iron, including dust, which is shown here in the blue. And it was interesting that the community in the transition zone, where we know there's a lot of dust input, the heterotrophic bacteria could grow very well on this dust. And in fact, they grew better on dust than some other iron sources that should be pretty easy for them to grow on. However, pico-eukaryotes, for example, could not grow very well in the dust, and they could grow much better just on simple inorganic iron, for example. So it suggests that bacteria might be really kind of primed to access this mineral form of iron, particularly in the transition zone. And this was interesting because we saw really high siderophore concentrations within the transition zone. And we had originally thought that perhaps microbes might be kind of primed to produce siderophores, particularly actually in the subpolar gyre, which is an HNLC region, so very low iron. And since we know that siderophore production in cultures, at least, is regulated by the ambient iron in the media, we thought that the ability or the metabolic potential to produce siderophores might be highest in kind of the lower iron regions of our transect. And in fact, that was not the case. So if we look at metagenomes, both from our first cruise and then our second cruise, we can see that the metabolic potential for both siderophore transport as well as siderophore synthesis was kind of widespread throughout all the latitudes that we sampled. And this is just in surface waters. And it was very consistent across latitude. And we also were able to identify many more siderophore transport genes compared to synthesis genes. So this suggests that some microbes might just have the ability to transport or take up the siderophores rather than to produce them themselves. If we dig a little bit deeper into metatranscriptomes, this tells us not only the metabolic potential for producing and consuming these compounds, but also gives us an idea of which microbes are actually turning on the genes at the time of our sampling for producing and consuming these compounds. And so this is just one kind of vignette of this, but these are three very similar siderophores that we identified in our cruises, and where they have the red dots is where they were found along the transect. And then this is showing the transcripts for the expression of ferrioxamine uptake genes by alpha proteobacteria, gamma proteobacteria, and others in the 0.2 micron size fraction. And we saw that kind of the expression of these ferrioxamine uptake genes really varied depending on the type of organisms you were looking at and also the type of siderophore. So there was not some, clear pattern where every organism was upregulating these siderophore uptake genes only in the transition zone or only in the HNLC environment. But in fact, it really varied depending on the compound or the type of bacteria that you were looking at. And so these together are telling us that kind of the use of these compounds is actually quite widespread, and it varies among organisms and is kind of going on in all kinds of environments in the ocean. And so with kind of all this data together, we've been thinking a little bit about the role that these compounds are playing in the environment. And we think that kind of siderophore production by heterotrophic bacteria is sort of fixing dust or mineral forms of iron for the rest of the microbial community to take up. So we could have dust that's delivered to surface waters, and then siderophores are released by the heterotrophic bacteria community. These siderophores bind the iron from the dust and kind of transfer it into the dissolved pool, where it can then be acquired by the producing bacterium, or it can be acquired by other microbes that might have the uptake genes for these siderophore iron complexes. But it's probably limited to only those that have those particular siderophore transport genes. But this might be kind of almost like a service to the community that heterotrophic bacteria are performing, where they're like fixing iron into this more usable form. And this is interesting because this also could be kind of selecting for particular microbes, again, that contain these siderophore transport genes. And I think this is one really nice, elegant example that Shane Hogel just published, where we contributed some of our siderophore data. And what he found was that if he looked in single-cell genomes for picocyanobacteria from the Atlantic, as well as the Pacific Ocean, he found that only those present in the Pacific had siderophore uptake genes. And he was able to identify for the first time these siderophore uptake genes in picocyanobacteria. And the Pacific is a much lower iron basin. That's what these background colors are showing. And so the fact that they were only present in the Pacific suggests that iron scarcity could be kind of selecting for this ability in picocyanobacteria. And so this is showing that there could be this really important control on both kind of iron availability and the chemical form of that iron on microbial community structure. Okay, so that was my kind of first surface ocean vignette. And so for the last bit of the talk, I'm going to kind of transition into the deep ocean, and we can think a little bit about how these ligands might be impacting iron in the deep ocean, particularly in hydrothermal environments. And this is work that is being done by Patrick Monreal, who's an undergrad that was working in our lab and is graduating from Stanford. And then Laura Moore, one of my graduate students, and then a postdoc, Colleen Hoffman. And we're interested in thinking about this particular aspect of iron cycling because hydrothermal iron is a big proportion of the dissolved iron that's in the deep ocean. And as I mentioned earlier, there was kind of this pretty surprising finding from geotraces where when we collected data for dissolved iron over the southern East Pacific rise, Ridgecrest, we saw this huge plume of dissolved iron that extended for greater than 4,000 kilometers. And that was pretty surprising because iron that is input into the deep ocean from hydrothermal vents is an inorganic iron form. And we know that inorganic iron is very insoluble. So when it mixes with oxygenated seawater, it was thought that pretty much all of it should just precipitate as particles right next to the vent site. And that is actually largely true, but a really small proportion of it stays in the dissolve phase. And because iron is so low in seawater in general, only a very small fraction of this iron actually has to remain in the dissolve phase for it to have kind of a pretty big impact and for us to be able to see this nice long plume. However, it was still very surprising that it persists for so many thousands of kilometers. And so this study was kind of complemented with some modeling work, which is shown here in this plot, where they were attempting to kind of manipulate different parameters in the model to kind of best match the distributions of dissolved iron coming from the ridge crest. So the observations are in these little diamonds, open diamonds here, and different model runs are in these colored lines. And this is showing kind of like going from the ridge crest on this side and then away from the ridge crest going to the left. And they modeled different amounts of iron and different amounts of ligands coming from the ridge crest. So they were kind of hypothesizing that organic ligands could be responsible for stabilizing this hydrothermal iron. But we weren't really sure what the ligand concentrations were like in these vents, or if siderophores are present, or what the characteristics of these compounds might be. And so my postdoc, Colleen Hoffman, she set out to kind of address this question, and we were meant to go and sample the southern East Pacific rise like 3 years ago, but then of course we couldn't because of the pandemic. And so we were only able to do this cruise pretty recently, this past fall, but I'll mention a little bit about that afterwards. But in the meantime, Colleen was able to get some samples from the Mid-Atlantic Ridge and the Mid-Cayman Rise from some very generous colleagues that helped her kind of pivot her postdoc in the meantime. And this is pretty neat because now we have samples from several different hydrothermal vents where we can look at ligands, and they kind of span a large range in spreading rates, going from kind of ultra slow spreading, and then fast spreading. And these kind of have a range of iron concentrations as well, with the faster spreading rates having much higher iron concentrations. They also have very different geochemistries and geological settings as well. So the first really interesting thing that Colleen found, this is just in the Mid-Atlantic Ridge data set, which I'm going to show you now, is she saw that strong ligands were present in all of these vents that she sampled along the Mid-Atlantic Ridge. And the strong ligands and dissolved iron had this like very tight one-to-one coupling throughout these neutrally buoyant plumes. And this was really interesting because it was all these samples are from the Mid-Atlantic Ridge, but it was many different vents that have very different characteristics, ranging from really high iron concentrations to quite low. And Lost City is a very unique, very different type of geological setting compared to all the other vents. And so it was cool that this relationship was kind of apparent across this range in dissolved iron concentration and vent geology types. So there was this kind of like very important role that these strong ligands were playing in this system. But because we're also interested in measuring siderophores, we were kind of wondering if these strong ligands are siderophores in this hydrothermal environment. And that might seem surprising because I was telling you how they seem to be mostly regulated by kind of low iron concentrations. But in fact, this particular study had looked for siderophore uptake and biosynthesis genes in a hydrothermal system from the Guaymas Basin, which is kind of a pretty different type of hydrothermal vent, but they saw transcripts that were pretty abundant pretty abundant transcripts that were accounting for both siderophore uptake, siderophore regulation, and siderophore synthesis in this system. But they didn't measure siderophores directly. They were just kind of looking at this from an omics perspective. But they were suggesting that siderophores were being used kind of in a similar way to what we've been talking about within surface waters, where they're using to kind of mobilize iron and disperse it throughout this plume by the microbes. So we, of course, wanted to be able to measure these, and that's what Colleen did. So she measured siderophores both along the mid-Atlantic ridge and from the mid-Cayman rise, and she found siderophores on every vent that we've sampled, which is really interesting. And she saw a wide variety of compounds. Many of them are pretty different than ones that we've seen in surface waters, but some of them are actually quite similar. So all of these rows are different siderophores that we identified at each of the sites. And then if it has a colored box, that means that it was present at that particular site. And these columns here are from lower iron kind of fracture zone type sites. And then the ones to the right are from the spreading centers along the mid-Atlantic ridge. And one thing that was interesting is that some of the compounds we found at many of the sites are these ones that have this kind of long fatty acid tail, and those are really common in surface water. So it's interesting that we found some similar compounds. And then this one, for example, vibrioferin, is one that we hadn't identified yet in seawater. So that was also pretty cool. And then we also found them at the mid-Cayman rise, and some of these compounds at the mid-Cayman rise were the same that we saw in the mid-Atlantic ridge as well. And this was data that the undergrad Patrick worked on. So this was pretty cool that we were able to measure these compounds for the first time in hydrothermal systems. And so what about these kind of other fast spreading ridges? These are really high iron ridges, and this is where I showed you that huge plume of dissolved iron. So we've been really curious if siderophores and/or strong ligands are present in that particular plume. And so that's what we went about sampling on the Plume Raiders cruise that Sarah mentioned. And this was our group, and it was a really fun, dynamic team. And it was a long cruise, but it was well worth it since we had been waiting several years in order to go out on this cruise. And so our kind of hypothesis for this expedition was that we thought that ligands might be being produced by microbial communities actually kind of surrounding the vents in these like diffuse vent sites that are sort of on the flanks of the ridge crest, and that they maybe are getting kind of entrained into the buoyant plume. So we set out to kind of sample a bunch of different high temperature and low temperature or diffuse flow type vents. And this is my data from my student, Laura Moore, and this is kind of hot off the press. She was doing some analyses while we were at sea, and then she's been hard at work since we got back. And this is showing some measurements that she made at sea using our voltammetry method on the smaller volume samples. And this is showing box plots of the strength of the ligands that she identified based on kind of clumping them by their height off the seafloor. So these ones are samples that fell within this kind of high temperature, neutrally buoyant plumes, and those contain the strongest ligands. So these are kind of binding strengths similar to siderophore type ligands. And then samples that fell outside of the plume had weaker ligands. So this suggests that the ligand composition is kind of variable depending on if you're inside or outside of the plume, and probably has unique sources. She also just recently got our first measurements of the concentrations of siderophores in these samples. And so I'm just showing example profiles from 3 different stations. And all of these stations had some diffuse venting near the seafloor, as well as a little bit of high temperature venting higher up in the profiles in these 2 stations. And what was really interesting is that we often saw the highest concentrations of siderophores at each of these stations near the seafloor, very close to these diffuse vents. So this is a picture that Century was able to get of one of our diffuse venting type sites. But we also saw high siderophore concentrations within the high temperature neutrally buoyant plumes as well, which are these 2 points here. And the concentrations are actually pretty high relatively, so up to kind of about 100 picomolar or so, which is a bit higher than what we've been seeing in surface waters. So this was really exciting to us so far, and we're really excited to kind of dig into this a bit more. So the kind of framework that we're thinking about right now in terms of these hydrothermal systems is that we think siderophores are probably contributing a lot to kind of exchange of dissolved and particulate iron in these neutrally buoyant plumes specifically, and perhaps are also kind of being produced near the seafloor in these diffuse vents and getting entrained into the neutrally buoyant plume. And the reason we're kind of thinking about that is because previous work, this is an example from Jess Fitzsimmons talking about this same hydrothermal plume from the Southern East Pacific rise, but she had her group had hypothesized that there's really rapid exchange between dissolved and particulate pools within this neutrally buoyant plume. And that's part of the reason it has such a long residence time in seawater. And so we think the siderophores are probably being used by microbes within the neutrally buoyant plume to kind of facilitate the exchange of iron between the particulate and dissolved phase. They can take it up for various metabolisms, but perhaps there's also some ligands that get kind of entrained into the plume initially as well. And this is interesting because although it's this high iron environment, we think that siderophores are being used in kind of a similar mechanism as we're seeing in surface waters, where they're kind of solubilizing iron from dust just to get at this extra iron source in surface waters. So to conclude, I showed you a couple of vignettes about how microbes are producing organic ligands in the marine environment. So we're thinking that a lot of heterotrophic marine bacteria are producing siderophores to transform particulate iron into more usable forms, both kind of for themselves, but also probably for the wider microbial community that can kind of steal these siderophore iron compounds. The ability to synthesize and take up siderophores seems to be very widespread in the ocean. It's not confined to iron-limited regions, and we're seeing more and more the ability to produce and use siderophores all over the ocean. So I think we're kind of shifting our paradigm about where these compounds are being produced. I also showed you that low iron availability can kind of impact microbial composition and selects for populations that perhaps can access the siderophore-bound iron. And we've had, I showed you one really nice example for picocyanobacteria, but we're kind of exploring this also with eukaryotes as well. So I think it's very interesting. And then finally, I kind of ended showing you that ligands are being produced also in the deep ocean in these high iron hydrothermal systems, and they're likely facilitating the transformation between particulate and dissolved iron pools, even in the deep ocean as well. So finally, I just want to acknowledge other members of the Bundy Lab that I didn't mention today, captain and crew of many of the cruises that we went on to collect samples for the vignettes that I showed you, and then some additional co-authors and collaborators that contributed to this work and who have done a lot for the lab. And we don't have a lot of good pictures of the lab during COVID, but we were able to have a little backyard hangout recently with some friends and the Bundy Lab. So I put that one here, but and that's it. And I'll be happy to take any questions. Thank you.
Thank you, Randie. That's awesome. So I guess I'm going to be kind of fielding the questions.
Great.
If they come in from Zoom or the chat. Also, we have a question from the audience. So Tom, if you can come up and ask her here. Thank you. She can hear you.
Okay, great talk, Randie.
Thanks. Hi, Tom.
I was interested in what the role of ligands might be in the coastal zone, like the California current. Would you still see a similar kind of one-to-one relationship there, or are you close enough to terrestrial sources where maybe the inorganic would be more important?
That's a really good question. We haven't looked at it that closely, but there's definitely still a strong relationship between the strong ligands and iron and like the California current, and even in estuaries. It's kind of like the iron that's able to actually escape estuaries like San Francisco Bay, for example, is like iron that's associated with strong ligands. So they tend to have this kind of like one-to-one relationship when kind of coming out of the estuary at like the high salinity end. And so I think there's actually a lot of similar dynamics going on that I was talking about in hydrothermal systems with even coastal environments, and kind of like upwelled iron from the benthic boundary layer and stuff like that. Yeah. good question.
Yeah, thank you.
Yeah.
So I have a question too.
Yeah.
So one question is how deliberate siderophore production is in order to mediate kind of microbial interactions, like certain bacterial taxa that might be making a siderophore that a certain type of phytoplankton might like.
Yeah.
Like that's a kind of a cool thing to consider. And I wonder if you would see or you expect to see differences in based on like surface siderophores from HNLC versus the plume where they wouldn't be making siderophores for phytoplankton.
Yeah, I think that's a really interesting question, and we haven't really looked at it, but I've been trying to think a little bit more about like the kind of phytoplankton bacteria interaction component. Like if there's model systems where there's like a nice mutualism going on where like the phytoplankton has like the siderophore transport genes that their associated bacteria is making, for example, or if there's probably some examples of that, but then I think there's probably also other examples of like competition for iron where, you know, the heterotrophs are making siderophores that the, you know, eukaryote can't take up, for example. But I suspect that there's probably, we're seeing like pretty different compounds in the hydrothermal environment. So yeah, I wonder if it is related to those kind of interactions. And I actually hadn't really thought about that before, but that's a really good idea.
So cool. And so cool to think about like identifying those interactions, but you can't do it unless you know what the siderophores are.
Yeah.
Right?
So I feel like, yeah, now we're getting to all these cool, like second order questions, but it took us so long just to identify what's out there. But
yeah, cool.
Yeah.
I have another question, but I don't want to hog it if there's. So, okay, so I'll ask my other question. So my other question is about like iron fertilization and whether you think an iron fertilization experiment with just the inorganic iron versus siderophore bound iron would have a different outcome in the community.
Yeah,
or which would be more effective.
Yeah, I think so. I think that in terms of iron fertilization, if you're adding a bunch of iron to seawater and you add it as an inorganic phase, you're going to immediately lose a bunch of it. So that's one aspect, just like kind of maximizing how much of it stays in the soluble phase, like adding it in some organic form would keep more of it soluble. So just keep more of it available. But we also know that when you add iron in like kind of either a natural or artificial iron fertilization scenario, that like the microbial community basically like immediately starts producing ligands for the same reason, probably to like try to, you know, keep most of it soluble. And then I think like in terms of choosing which organic form of iron you would add, I think that's the tricky part because we just still don't know that much about just kind of like the transport genes for different organic iron complexes. And I talked a lot about siderophores, but we're also missing like a huge proportion of the, you know, we haven't been able to characterize like basically 90% of the organic iron pool. And so we don't know what that is. So it's hard to know, you know, which microbes have the transporters for those things. And, you know, if most of it's in humic substances or something else, which I think would be important to know.
Cool. Yeah, lots of research to do.
Yeah.
So I don't see any questions in the chat. Does anyone in the room have a question? Well, if there are no more questions, please join me in thanking Dr. Bundy again for a wonderful presentation.
Thank you so much. Thanks for having me.
Hope to have you here in person before too long.
Yeah, that would be great.